Hello, and thank you for joining us for this exciting virtual session on bioinformatics and artificial intelligence for cancer research and development. We hope you're all staying safe and well. Thanks first to the incredible AACR committee and organisers for making this possible in such exceptional circumstances. My name is Jonathan Dry. Until recently, I was Director of Bioinformatics and Data Science within Oncology Research and Early Development at AstraZeneca, where I was also helping shape the AI transformation. But more recently, I've moved to Tempus as VP of Scientific Discovery, where we're matching clinical to molecular data for patients with cancer and other diseases, and pairing this to technology that enables physicians to make real-time personalized decisions for patient care and driving therapeutic discovery, development, and delivery. And I'm delighted to be joined by Dr. Hannah Carter, Assistant Professor in the UCSD Department of Medicine, where she's working on cancer bioinformatics with a focus on variant interpretation and cancer precision medicine. So to quote Sydney Brenner, progress in science depends on new techniques, new discoveries, new ideas, probably in that order. And the last couple of years have seen several major breakthroughs in new techniques, bringing data of an unprecedented depth, structuring health records and epidemiological data, convergence of multimodal patient data, epigenomic and multiomic platforms, and pairing of germline to somatic genomics, longitudinal omics from liquid biopsies, plus evolutions in disease models and model systems like organoids fueling new dimensions of phenomics and CRISPR and dynamic omics. And all this is becoming possible at a single cell and spatially resolved resolution. Thankfully, recent advances in AI have brought powerful new tools and techniques into the computational biologist toolkit, able to handle multimodal networks and graphs. But these techniques in themselves have also opened up the possibility to harness an omic level richness of data from imaging, video, text, knowledge, and data archives. So now more than ever, computational techniques are at the forefront of our ability to drive discoveries and ideas from these data to help cancer patients. We have an opportunity to understand our patients at a systems level and a cellular resolution, to create a Google map of a patient or a tumor with which to direct optimal therapy and new discovery. It's been great to see how quickly these capabilities can now be mobilized to accelerate research and aid action for the current pandemic. And we hope this momentum can be carried into our cancer research. So I'd like to thank all the authors that sent such incredible abstracts to the meeting. We hope we've selected talks for this session that show the impact of bioinformatics and data sciences throughout the journey from discovery to the patient, including innovative approaches towards disease detection and diagnosis, detection of targetable disease drivers and dependencies, elucidation of drug and resistance mechanisms, and steering trial matching and decision support. Our first video in the session is going to be from Xingyi Shi, a PhD candidate in the bioinformatics program at Boston University. She'll be presenting their single-cell profiling of bronchial brushings to identify detectable epithelial and immune cell signatures indicative of underlying lung cancer subtypes. So over to our first video.
Good morning. My name is Xingyi Shi. I am currently a PhD candidate at Boston University Computational Biomedicine section. My presentation today is on non-small cell lung cancer subtype-associated molecular and cellular alterations in the normal appearing bronchial epithelium. Here is my disclosure. Molecular changes in the airway are resulted from an etiologic field of injury and field of cancerization. The etiologic field of injury reflects the aberrant physiological response to carcinogen exposure that creates a susceptible microenvironment for cancer initiation. In contrast, field cancerization reflects the effects of first-hit mutation in a clone of cells from which the tumor ultimately arises. In the airway of a patient developing cancer, there is an interplay of these two aforementioned fields affecting the molecular makeup. Cancer-associated gene expression changes in the bronchial airway have recently been used to develop and validate a 23-gene classifier called Percepta that is currently being used clinically to improve the diagnostic yield of bronchoscopy for lung cancer among patients with indeterminate bronchoscopy. The AEGIS trial collected samples from high-risk patients under suspicion for lung cancer, and the performance of the biomarker was evaluated in the subset of those patients with non-diagnostic bronchoscopies. Given that our previous cancer genomic classifier, Percepta, was developed in a NSCLC subtype agnostic fashion, more work is necessary to explore additional transcriptomic changes that can discriminate between lung squamous cell carcinoma and lung adenocarcinoma for targeted diagnosis and precision medicine for treatment. While responses to smoking may result in different sets of changes and different field of injury that predispose the airway to develop adeno versus squamous cell carcinoma, field cancerization may also be different due to the cell of origin and location distribution differences. In this project, we're investigating the transcriptomic changes associated with NSCLC subtypes in the normal appearing airway by first looking into the bulk gene expression differences between squamous and adenocarcinoma patients, and second, the distinct cell types involved in the origin or progression, respectively. Shown here are the data sets we used and the analysis design. On the right-hand side, we first used the same cohort of samples as Percepta biomarker to derive a bulk squamous versus adenocarcinoma signature. In parallel, shown on the left-hand side, we profiled cells from normal appearing airway epithelium from 17 ever smoker patients also undergoing bronchoscopy for the suspicion of lung cancer using a plate-based single-cell RNA-seq method under a DoD-funded cohort. With transcriptomic information from both the bulk and single-cell resolution, we are able to estimate the cell-level contribution to the overall molecular differences detected between adeno and squamous patients that are enabling the disease progression. From the bulk transcriptomics of more than 300 NSCLC patients, we identified 247 genes upregulated in the adeno subtype. Their functions are enriched for cilium-related functions. 204 genes identified to be upregulated in squamous cell carcinoma subtype, and their functions are enriched for pathways related to TNF-alpha, NF-kappa B signaling, and keratinization. These two signatures can be replicated in a separate validation set, AEGIS-2 cohort, with about 200 samples and significant p-values. Notably, given that the lung squamous cell carcinoma often appears in proximal airways, our squamous signature was also enriched for another airway signature associated with proliferative lung squamous pre-malignant lesion from an independent cohort. The airway proliferative subtype signal is derived from our previously published datasets. It is essentially the gene expression alterations detected in the normal appearing large airways that are associated with disease severity and histological grade of squamous cell carcinoma pre-malignant lesion. While our current squamous cell carcinoma signature is significantly and concordantly enriched in the proliferative lesion signal shown in the GSEA figures here, it reflects that the NSCLC subtype associated alterations may exist prior to the development of lung cancer in normal appearing airways. Meanwhile, we were also interested to find out what cell types are involved in NSCLC specific molecular changes. Using single-cell transcriptomics, we profiled about 2,000 cells from 17 patients under the same clinical condition, and we identified 15 cell types, including the novel ionocytes in normal appearing bronchial epithelium. WGCNA algorithm was utilized to find clusters of co-expressed genes across the single-cell RNA-seq gene expression matrix to examine genes in a functional and co-expressed gene modules. 20 gene modules were thus identified using unsupervised approach, and the gene modules are shown to express in a cell type specific manner. Upon examining the NSCLC signature broken down into the cell type associated gene modules, we notice that the adeno signature labeled with the red bar is enriched for a ciliated cell module and a novel ionocyte module. On the other hand, the squamous signature marked in blue bar is largely consisted of 2 gene modules associated with secretory cells. One-sided Fisher's exact tests were conducted to conclude the significance of these enrichments in specific NACLC signatures. Among them, the ionocyte gene module has overall metagene score significantly different between adeno and squamous subtypes, suggesting a potential novel function of ionocyte in shaping the NACLC molecular compositions. These results show that the epithelial cells, especially secretory, ciliated, and ionocyte populations, contribute differently towards the adenocarcinoma versus squamous cell carcinoma in normal airway epithelium. We then compare the NSCLC patients to patients with benign diseases in the same Aegis cohort, and we identified 4 out of 20 gene modules showing significantly different metagene score between them, with stringent p-value cutoff and replicable direction match in Aegis 2 cohort. Specifically, a gene module associated with neutrophil and macrophage functions has metagene scores significantly different between benign and all the NACLC subtypes. A gene module associated with dendritic cell function, a gene module associated with a gene module bridged between secretory cells and dendritic cells, and the previous one specific to ionocyte are significantly different between benign and squamous cell carcinoma only. These results show distinctive immune cell contribution towards lung adenocarcinoma and lung squamous cell carcinoma when compared with benign diseases. To conclude, we identified lung squamous cell carcinoma specific molecular differences in the airway field of injury with squamous versus adeno that are highly expressed in secretory, ciliated, and ionocyte cell populations, indicative of a squamous-like epithelium. When compared to benign lung diseases, we identified NACLC shared molecular differences in myeloid immune cells and squamous cell carcinoma specific molecular changes in dendritic cells. Our results suggest that there are different responses to injury in the normal appearing airway among NSCLC subtypes, which shed light on the molecular and cellular foundation of the diseases. These results might be helpful for the design of targeted therapies and for directing screening regimen among NSCLC subtypes. Thank you, and here is my acknowledgement.
Okay, we now have some time for questions for Dr. Shi. I don't see any in the Slido at this time. So if you have questions, please type your questions. Okay, Dr. Shi, do you see a question there? So the question to Dr. Shi is, have comparisons been made to the circulating tumor DNA for potential detect cancers early?
Yes, thank you. That's a very interesting question, but we haven't done that. It's very interesting field to look into ctDNA right now, but that requires very deep sequencing depth, and with single cells, it's hard to sequencing and detect the circulating tumor DNAs for now. We haven't looked into that, but that'll be definitely interesting project to look into.
And in your analysis, did you see any association with sex or age?
In this cohort, we have more male and more male and older people in our cohort so far because of the setting of the clinical setting. But we do not, even though it's a very limited range, we do not see age association and sex differences in our data set.
Next question. Can you use any of these new signatures as a classifier between lung adenocarcinoma and lung squamous cell cancer? It looks as if they overlap.
Not sure the second part, but for the signatures, I think that was our original idea, was that using single cell, the clinical application of this project was to potentially find a target markers for biomarker development to differentiate lung adeno and squamous subtypes so that we can have different screening cohort or different treatment therapies. So that's one potential application of the project. I don't think they overlap.
Yeah. Did you see any association with race in your cohort?
We didn't look into the race because this project is very biased towards the white people. So we don't have enough power to look into race.
Okay. Can the bulk RNA sequencing information be replaced by the single cell RNA-seq measurements averaged?
Yes, we did that sanity check by combining all the counts from the single cell data to create a pseudo bulk count matrix, and that the correlation, the signal largely match up.
Was there any indication of immune fitness and immune cell exhaustion detectable in the bronchial brushing single cells that could direct immunotherapy?
That's an interesting question. I think one of the findings we report was that there is differences in the myeloid cells between benign and NSCLC patients. And so that could potentially be used to direct immunotherapy targeting those cell types, given that they are the targeted cell types.
No, there's a question about what type of classifier was used.
Oh, sorry. I think okay. If I understand it correctly, we're using generalized linear model.
Great. And there's a question about when the data set will be available.
We're currently working on the manuscript, so I hopefully within this year we'll have the data set available.
Fantastic. At this time, it looks like there are no further questions.
Okay, thank you.
Oh, one last question popped in. I think we still have a minute. So,
yeah, that's a good question to follow up on. So I guess we do report some of the squamous specific signatures, and we could potentially use that for differentiating the non-small cell lung cancer subtypes. And we also report pan NSCLC differences compared to the benign. And so that's two classifier or two directions we're looking into.
Okay, great. So I think we're now at the time for the second talk. So I'm pleased to introduce Dr. Ruzbe Dehanasiri, who is a postdoctoral research fellow at the biochemistry department at Stanford School of Medicine. He's going to be presenting a method called Sicilian that is capable of calling fusions in single cell sequencing data and will describe applying this to assess fusion heterogeneity in cancer samples.
My name is Ruzbe Dehganesseri. I'm a postdoc at Stanford University School of Medicine and Department of Biochemistry. The title of my talk is Sicilian, Precise and Unbiased Detection of Gene Fusions at the Resolution of Single Cells Using Improved Statistical Modeling. I have no financial disclosure. Gene fusions are known to drive many cancers and they can reveal critical insights into carcinogenesis. And because they are absent in normal tissues, they are among the most powerful biomarkers and therapeutic targets. Perhaps the most famous fusion is BCR-ABL1, which is a characteristic of chronic myeloid leukemia, and this fusion has led to the discovery of imatinib in 1992, which highlights the clinical significance of gene fusions. As we are living in the era of high-throughput next-gen sequencing technologies, we have the unique opportunity of using hundreds of publicly available massive sequencing databases to have accelerated discovery of gene fusions and RNA variants in general by using precise computational methods. However, despite this great promise, there are still many aspects of gene fusions that are still unknown, and the main reason for that is that the current method still suffers from high false positives and false negatives. Single-cell RNA-seq provides molecular profiling of single cells and can inform how RNA splicing and gene fusions can control gene expression levels in a single cell. A precondition to this promise is to have a precise computational method for quantification of RNA variants in single cells. However, RNA splicing detection in single cells is extremely challenging due to many single cell inherent biochemical and technical errors, such as dropouts, small amount of transcriptomic information, substantial sequencing noise, and computational biases in the alignment. To address these challenges, we have developed Sicilian, which is a statistical wrapper that can be implemented on top of any conventional splice-aware aligner and can be used to basically detect all sorts of RNA variants in single cells, such as RNA splicing junction, gene fusions, and circular RNA. Here is the summary of Sicilian workflow for splicing junction detection. We begin from an alignment file from the splice aligner, which is usually in the format of a BAM file, and our goal is to find a refined list of true positive splice junction out of that BAM file. So the first step in Sicilian is to first build a database of candidate splice junction out of that BAM file by basically extracting all junctional alignment information. And then Sicilian employs its statistical modeling to assign a statistical confidence to each junction within that database. And here is the very big picture of the statistical modeling in Sicilian. We first assign a read level score to each junctional read alignment, and we basically use a general linear model, and the predictors in the model are various alignment features such as alignment score, mapping quality, number of mismatches, and read entropy. And then for each junction, we basically aggregate all the read level scores across all the reads that have been aligned to that junction to basically find an aggregated junction score for that junction, which is basically an empirical p-value. And then we use a cutoff for the empirical p-values and call junctions by basically considering a threshold. One of the challenges of single-cell analysis is the increased false positive rate due to multiple hypothesis testing of every junction across thousands of single cells and samples. To address this issue, in Sicilian, we adopt a multiple hypothesis testing correction strategy in which for each junction we basically collect the empirical p-value across all samples and then compute the median of those empirical p-values as a unified criterion to decide whether the junction should be called across all cells or not. This median score is basically the final score by Sicilian, and we call it Sicilian score. To benchmark Sicilian's performance for splicing junction detection, we have used matched single cell and bulk cell lines from a recent single cell benchmark study in which all single cells in the 10x sample come from 5 human lung adenocarcinoma cell lines, and therefore we can use bulk cell lines as genomic ground truth for single cells. We have basically run Sicilian on both single cell and bulk data sets, and we compared its performance against the case that we use the number of reads for the junctions as the criteria for calling junctions. And we can see that by using Sicilian, the concordance between the detected junctions on single cells and their corresponding original bulk cell line have increased. For example, for cell line HCCH27, we can see that while based on Sicilian, around 74% of junctions in the single cells have been also detected in the bulk HCCH27 cell line. Based on read count criteria, that fraction is much lower. It is around 50%. So we can see that by using Sicilian, we have been able to basically improve the concordance between single cells and their corresponding bulk cell line. We have also applied Sicilian to human lung cell atlas dataset, which is a single cell atlas of human lung cells in which more than 75,000 cells have been profiled using both 10x and SMART-SIG2 protocols. As there is no ground truth for these datasets, we have basically used annotated junctions as targets for true positives and false unannotated junctions as targets for false positive junctions. And by this definition of true positive and false positive junction, we can basically obtain the ROC curves based on Sicilian and read count based criteria. And we can see that the AUC by Sicilian is significantly higher than the AUC by read count. We have further compared Sicilian performance on the HLCA dataset by comparing its detected junctions with two of the most recent databases of non-human splice junctions, GTEC and CHESS. And we can see that while before Sicilian only around 10% of junctions were shared with those databases, after Sicilian around 50% of junctions are shared with those databases, which highlights that Sicilian in an unbiased way can enrich for non-splice junctions. For chimeric detection using Sicilian, we basically use chimeric alignments from a star and build a database of candidate fusions. And then we use the statistical model that we had trained for splicing junctions to make the initial predictions on candidate fusions. And then by looking at the overall distribution of statistical scores, by this model across all candidate fusions, we basically select those fusions that have extremely low and extremely high scores as negative and positive training data to then train a chimeric specific model, which can be used in the second step to make the final predictions on the fusions in the database to obtain a statistical score for each one of them. And then by looking at those statistical scores, we can basically obtain the final list of called fusions. And the main reason for having this two-step approach for fusion junctions is that some of the alignment features for chimeric are not available for chimeric alignments, and also we need to have a definition criteria for having training data for chimeric junctions. To evaluate the performance of Sicilian for fusion detection, we have compared it with star fusion, which is another wrapper for fusion detection using chimeric alignment in a star. And we use two simulated data sets that have 500 true positive fusions. And we can see that while Sicilian reports slightly fewer true positives than star fusion, it it has better specificity and reports fewer false positives than star fusion. So for example, for one of the data sets, it reports 7 fewer true positives, but on the other true positive, but on the other hand, also reports 2 fewer false positives. So while its sensitivity is comparable, its specificity is much better than star fusion. And here we can see the ROC curve by Sicilian on one of the data sets and its AUC value, which is extremely close to 1. To conclude my talk, we have seen that Sicilian enables systematic profiling of gene fusions and RNA isoforms at the resolution of single cells. It provides an unbiased statistical framework for discriminating false positives from true positives, and we are currently collaborating with several single-cell consortia at Stanford to build a comprehensive splicing map of normal and tumor cells. With that, I would like to thank our members in the Salzman Lab, especially Julia Salzman and also Julia Olivieri, who has been working with me on this project. And I would also like to acknowledge our funding for this project. And thank you so much for your attention to my talk, and I would be happy to take any questions. Thank you.
Thank you, Dr. Ganastri, for that fantastic pre-recorded talk. I can see we've got some questions coming through to the chat at the moment. So kicking off with more of a technical question. So could you describe a little bit more about what variables most inform the detection in the GLM model?
Yes, sure. So basically in the GLM model, so that we use for basically training the model for discriminating false positives and true positive junctions, we use various alignment features such as alignment score, mapping qualities, number of mismatches, and read entropies. And basically, I would say that mapping quality had a very strong effect on basically discriminating false positives and true positives. And basically, most of the junctions that had multi-mapping reads, they were mostly false positives. But basically most of the alignment features had a significant contribution to the eventual score for each junction. And basically, and also we noticed that most of the junctions that had been also aligned to their reference genome, they were mostly false positives.
Great, thank you. A question about the single cell resolution. So did you first look at the differences in fusions and splicing detected in the cancer cells versus any stroma or immune cells across your single cell population?
Yes, that's a great question. Actually, we haven't done like a comprehensive profiling of gene fusions at the moment. We are still working on basically running our pipeline on the single cell data that we have obtained through our collaborations, and that's an exciting question to answer through running this pipeline, and we are currently working on that to basically see what are the cell type specific behavior of gene fusions and how they can control gene expression and other splicing events at the single cell resolution.
Great. And I guess it might be the same answer then, but similar question, but more asking about the heterogeneity that you observed across the cancer cells.
Sure. So again, we haven't gotten the final results based on our pipeline on real single cell cancer data sets, but based on the previous study that we had done on gene fusions for bulk RNA-seq cancer data sets, we had observed extensive and substantial heterogeneity for fusions across various cancers. So some cancers such as prostate adenocarcinoma had much higher abundance of gene fusions, for example.
Great. A couple of questions relating to the depth of sequencing that you get from the single cell data and whether that can drive some of the differences in detection between single cell and bulk.
Sure. So basically, yeah. So one of the issues that we have in single cell data is like the low amount of information that we have in each cell, and also the issue of dropouts that we might have in the single cell data. And To address that, we basically resorted to statistical modeling and basically using transcriptomic information across various single cells. So by using information across various cells, we can basically address the low sequencing depth issue in single cell data. And we had addressed that issue at 2 different levels. First, in the statistical modeling, in which we use alignment features and alignment information across various cells. And then in the second step, where we basically correct the final statistical score for each junction by looking at the distribution of the scores for that junction across various cells. And by that, we can basically address the increased false discovery rate due to multiple hypothesis testing issue. So yes, sequencing depth is an issue in single cell data. And to address that, we try to basically develop a statistical model to basically address this challenge in single cell data.
Great. Thank you very much. So there's a couple of other questions which I'll promote into the chat, and maybe you can answer them offline, but I think it's time to move to our next video now, so thank you for that. So I'm delighted to introduce our next speaker, Dr. Mika Adesmaki, who's Associate Director of Bioinformatics at AstraZeneca, where he shares an affiliation with Cancer Research UK, with whom they're developing a functional genomics centre in Cambridge, UK. He'll be presenting their approach to determine actionable genetic events and use machine learning to map these to the CRISPR and pharmacological data, screening data, to detect lung cancer cell dependencies. So over to Dr. Adesmaki's video.
Hello, everyone. My name is Mika Adesmaki, and I wanted to thank you for joining me for the next 10 minutes to hear more about how we are looking at novel targets by combining line of sight from the TCGA to CRISPR data in cell lines. So in AstraZeneca, we are looking at functional genomics from multiple different perspectives. So we are, of course, looking at public data sets like those from the Cancer Dependency Map and information from the Cancer Genome Atlas. But we also have an internal capability in what we call the Functional Genomics Center, where we're looking at pooled and arrayed CRISPR screens in treated cell lines for persistence and sensitivity. We also use AI and knowledge graphs to overlay the world's knowledge and information on all the inference we do in these data sets. However, today my focus will be primarily on the public data sets and looking at novel potential synthetic lethal targets. So what we've done in this analysis, we mapped the clinically prevalent cancer alteration of subtypes per tumor type from TCGA to cell lines. We then looked at the CRISPR data for these cell lines from the Broad Institute and Sanger Institute, and each of the subtypes we identify what are the CRISPR dependencies in the altered versus the wild type cell lines to yield potential synthetic lethal targets. So the different kinds of molecular alterations we're looking at, we first look at tumor-specific cancer genes, and we define these significantly mutated genes from different databases across 34 different tumor types. We further drill down into driver mutations based on hotspot mutation in the patient that's available. We also looked at copy number in tumor-specific recurrent regions that are gained or lost in 27 different tumor types. We also looked at cancer signaling pathways and did what we call a collapse analysis. So in some instances, a single gene may not be mutated more than once into the cell lines, but when we look at all the genes in the pathway, we might have multiple genes that have gain or loss of functions, and we could collapse these into what we call the 11 cancer pathways that we looked at. We also looked at clinical features like hormone receptor status and the microsatellite stability status for cancer subtype analysis, and all this in an effort to look for new novel targets. So how does it look like? So the subtypes, when we map them to cell lines, they provide what we call the binary event matrices, and there's an example here in breast cancer where we have a clinical prevalence for each of these alterations that are on the left, also given in the first column. And then for each of these cell lines, we give an indicator of 1 or 0 whether we have an alteration or. So for example, on the first row, we have p53 mutations, and the cell lines that have the also function p53 mutation will have a 1, otherwise they would be 0. And all of this we can then be used in our search for significant association with the map gene essentiality data, or for example, drug response data. We also wanted to look at this, how representative the cell lines are compared to clinical samples. So we looked at the frequencies of the alterations, and we found them to be fairly concordant. However, in some instances, like TP53, what we saw was that often in cell lines, P53 is more prevalent than is actually in the clinical settings. The same was for FAT1 copy number alterations. So we tied all of this together then in an internal app and visualization interface where the user firstly chooses a tumor type. In this case, it was, for example, colorectal cancer. Then they would choose a molecular alteration. And for this tumor type, we would have the clinical frequencies plotted for all the different kinds of alterations you can choose. And we've shortlisted. In this case, for example, microsatellite stability. And then at the bottom, we have a list of the top dependencies that are differential between the wild type and the altered lines. So in this case, unexpectedly, we get Werner helicase as the top hit when we compare MSI altered versus MSI wild type cancer cell lines. And in the same interface, we also have a volcano plot showing what's the mean difference between the altered and the wild type cell lines on the x-axis and the log 10p, negative log p-value on the y-axis. For each of these alterations that are then chosen, or CRISPR dependencies that are chosen, we also do a plot for each of the cell line values of the gene effect, where the more negative gene effects are where the knockout is causing an effect in the cells. So we want the altered cell lines to be considerably more negative than the wild type cell lines. Another example here in breast cancer, and looking at, for example, ER-positive breast cancers versus ER-negative breast cancers. And unsurprisingly, the top hits are ESR1, but also we get hits in related pathways like GATA3, CDK4, and FOXA1. That nicely shows the utility of this app in pulling out known associations. I'm going to now do a very short live demo of the app itself. So in this instance, I'm going to choose from the pull-down list from squamous. And having chosen that, we then get a plot, a bar chart of the different kinds of alterations and their frequencies. So we can look at, for example, CDKN2A alterations. Their clinical frequency is around 15%, but the cell line frequency is about 43%. So we choose CDKN2A. We can see that in total we have 19 cell lines with CRISPR data, and the first 8 have a CDKN2A mutation. We can see that the top hit is a gene called CEBPB. And in this instance, this gene, when we click on it, we see that it is the values for the altered cell lines are more negative in general than the values for the wild type cells. And in fact, CEBPB is known to be in same pathways as CDKN2A itself. So this is a potential target. And then looking at further evidence, for example, in CanSAR Black, we can see that there are 28 genes with ligandable cavities identified. So this is a potential target. And that concludes my presentation. So what we did, we developed an approach to draw a line of sight from TCGA, the tumor type, and map it on the Sanger and CCLA cell lines. And then we looked at the CRISPR dependencies and associated these to the line of sight molecular alterations to yield rational targets for bench follow-up. And we are currently investigating multiple of these in-house, so stay tuned. I wanted to also thank my co-contributors, and thank you for listening to my presentation.
Okay, great. We'll move on to the question and answer for Dr. Adesmaki. The first question we have is that high throughput screens like CRISPR will often give a long list of hits. What are your recommended steps to triage and prioritize the most actionable hits?
Yeah, thank you for the question. When we do get these lists, we're not looking at just in AstraZeneca just at the statistical significance, but adding other factors on top of that. So this could be something like are there ligands available, predicted ligands available within the targets, or are they essential genes, core essential genes, so likely to have issues with toxicity if molecules were designed against that. And generally using, I mentioned knowledge graphs, so if we're trying to overlay, for example, literature mining to see if the what kind of information the world has already around the targets that, and then re-rank it potentially around these multiple different criteria.
Great. We have a couple questions about whether the tools presented will be freely available to the public.
Right, so we are looking to publish this, not just the result, but we're also looking to make the app that we developed, the Shiny app, to the public, probably towards the end of the year, or maybe potentially next year. This will then, ideally, we would like to give people the opportunity to as well customize. So either they can download the app and deploy it internally if they want to look at different subtypes that we haven't thought about yet. So all of that is on the cards. So we very much like to do that towards the end of the year, like I mentioned. So stay tuned.
We have a question about future directions. I think you mentioned that you'll be testing some of the hits.
Yeah, so I haven't shown, of course, all the hits here that we found, and we have indeed plans to test some of these hypotheses we're generating through the app in the lab further. So either iterate CRISPR screens or potentially creating some isogenic models to give us a bit more cell lines. So for a lot of these molecular subtypes we're talking about maybe 3-4 altered cell lines. So we would like to get, of course, more confidence in that what we are finding is not just a chance finding in this data set.
What type of features are most influential in the machine learning to infer dependencies?
So in this instance, it's a very much we get the class labels from, we choose a tumor type, and the class label comes from whether the cell lines are altered for a subtype or not. And then into the machinery we feed the CRISPR data for all the genes so 16,000 plus genes that we have available from the Broad Institute's data. And then it's not super complicated, but it's like a linear model, basically a generalized linear model like algorithm that we're using here to rank the features in the first instance. So we thought about then if we could use these to build actually predictive models. So at the moment we're looking at associations and not predictive models. So we thought about building predictive models and seeing if we could say take the Broad Institute's data and then see if the predictions, we could build a predictive model and see if that holds in the Sanger data. But we found that we have too few samples really to do that, and there may be some batch effects in the Sanger data. So, but that's still part of maybe the future work that we're thinking about. But the short answer is that the features are just the genes in the CRISPR data.
Are you planning on adding transcriptomic data to the genomic features?
So adding transcriptomic data, the features, we'd have to think about where that makes the most sense. So a lot of our follow-up analysis would be to see if a target that we have identified is also expressed in the cell lines. So we've seen some instances where we thought we identified something interesting, but when we looked at the baseline expression in the cell lines in the RNA data that's available from the Broad Institute as well, there's no expression. So it could be that those were some chance and fluke findings that we just happen to be in the limited set of CRISPR data we have. So that's one way we've looked into as a sort of next step to using the RNA data, expression data.
Okay, one last question. Can you look at whether a combination of features, for example, CDKN2A mutation and KRAS mutation, better explain the DepMap essentiality?
Yes, that's an excellent question. So looking at these two things at the time. So the issue there is we are quite limited when we start looking at the subtypes. We're quite limited with the sample number. So if we tried every subtype versus every other subtype, we would have a combinatorial explosion there. But absolutely for the top hits we get, we absolutely have to have a follow-up step of seeing, is there another event that could explain some of the sort of lines that may not be dependent or maybe more dependent than other lines. So absolutely that we're kind of looking at using that as a sort of second step, but not for absolutely all the different possible combinations.
Great. Thank you, Dr. Adesmaki.
Thank you.
At this time, I'll introduce Mr. Jung-Hook Jeon, who's a graduate student in the Department of Chemical and Biomolecular Engineering at the Korea Advanced Institute of Science and Technology, and will be presenting a deep learning approach to predict the directional effect of a compound on gene expression in a cancer cell. We can start the video.
Hello everyone, I'm Junhyuk Jeon. I'm currently a graduate student in the Department of Chemical and Biomolecular Engineering, KAIST, South Korea. I'm working on the analysis of biological and chemical data using machine learning for my graduate studies. First, I'd like to thank the AACR organizers for this great opportunity to present my work. It is very unfortunate that we get to deliver talks online. I hope that everyone is fine. In this study, I will talk about deep learning models that predict upregulation and downregulation of genes in 8 different cancer cell lines in response to small molecules. Before we get started, I have nothing to disclose. Transcriptome landscape of cells upon a specific perturbation has been one of the most important indicators for understanding the cell's response to a compound in drug discovery. Therefore, transcriptome profiling has been often conducted to understand changes in gene expression levels and identify potential drug targets during drug development for various diseases. In this regard, L1000 was recently developed, which is a highly cost-effective transcriptome profiling method. L1000 can infer the expression levels of more than 9,000 genes with high reliability on the basis of the directly measured expression levels of a small number of genes called landmark genes. Again, for the remaining genes that are about 9,000, their expression levels are inferred based on the physically measured landmark genes. Thanks to this cost-effective technology, L1000 now provides over 1.3 million gene expression profiles. This large volume of dataset is an ideal ingredient for deep learning studies. Deep learning is a type of machine learning methods that can identify important features from an input by training the model using a big dataset and has been increasingly applied across various scientific and technical fields thanks to its highly accurate prediction performance. In this study, we developed a deep learning model named DeepCGR that predicts the effects of a compound on gene regulation by using L1000 profiles for the model training. Here, CGR stands for Compounds on Gene Regulation. DeepCGR was specifically developed for the 8 different cancer cell lines in this study and requires 2 different inputs, a protein sequence for which the regulation is to be predicted, and structural information of a compound of interest. Protein sequence and the structural information, given in a chemical language format called SMILES, are processed using convolutional neural network and multi-layer perceptron respectively. The outputs of the CNN and MLP are further processed by another MLP to generate a final outcome for a target protein, classifying whether the input protein is upregulated or downregulated in response to an input chemical. CNN was used among other deep learning methods for a protein sequence input because of its proven outstanding performance in processing a biological sequence. Also, we use the protein sequence for this modeling because a protein sequence as an input allows more streamlined processing of relevant biological information without the knowledge of this protein's function. Protein sequences have been used as an input for deep learning studies to predict various chemical and biological properties, including drug-target interactions, enzyme commission numbers, and protein-protein interactions. These studies show that protein sequence-based deep learning models not only performed well, but also can be applied to all kinds of protein sequences without relying on functional annotations such as locus tags or geo terms, which are sometimes not accurate enough. This is an overall scheme of DeepCGR. Again, smiles of an input compound is given to the model along with the protein sequence corresponding to each gene, and outputs are generated, classifying whether the given compound significantly regulates the gene. This scheme can be repeated for a large number of compounds for an entire set of genes in a cell. For training our model, we use the most pre-processed dataset called Level 5 dataset that covers the standardized gene expression levels. 1,000 profiles of the 8 cancer cell lines obtained at 10 micromole for each perturbation, and 24 hours were used for the model training, validation, and testing. We used only landmark genes with a reference protein sequence length of 1,000 or less, considering the trade-off between the model accuracy and the computational cost. Finally, upregulated and downregulated genes in the 1,000 profiles were labeled to train the DeepCGR. Genes were sorted in decreasing order of their expression levels, and the top and bottom 5% genes were selected for the upregulation and downregulation respectively. So, this is a slide that shows the prediction performance of the deep learning models developed for the 8 cancer cell lines. The average accuracy and AUCs appear to be good across all the cancer cell lines. It should be noted that this version of DeepCGR was trained with reference protein sequences from a metabolic database, CAG, for which cancer cell line specific mutations were not considered. At this point, we thought that the model performance could be further improved if the cancer cell line specific mutations were introduced to the reference sequences. Thus, we accordingly introduced the somatic mutation information available at COSMIC, the catalog of somatic mutations in cancer, to create another version of the DeepCGR with cancer cell line specific mutations considered. This slide shows the performance of the 2 different versions of DeepCGR, one with reference sequences and the other one with sequences with mutations. Contrary to our initial expectation, applying cell line specific mutations did not lead to the significant changes in the prediction performance. We suspect that this was largely due to a small number of mutations introduced compared to the total number of genes in a cancer cell line. We further evaluated the performance of DeepCGR for only the gene that has never appeared in the training set. For this, genes other than the landmark genes were also prepared by processing relevant protein sequences. Please note that the predictions for the downregulations were reasonably accurate, while the predictions for the upregulations needed further improvement. At the moment, we are not 100% sure regarding this observation, but in general, it is considered more difficult to define the effects of gene upregulations than the downregulation. Finally, we closely reviewed the classification results of DeepCGR, and we found that a particular chemical structure having a ring form was consistently observed for true negative predictions for the up and down regulations in the PC3 cell lines. Interestingly, similar observations were made for another prostate cancer cell line, VCAP. This sort of consistent observations suggest that we can further dig into the relationship between chemical structures and the resulting regulation outcomes, and possibly further infer regulation mechanisms involved. In this study, we developed the deep learning-based DeepCGR to predict the effects of a given compound on the regulation of a gene of interest by using protein sequence and chemical structure as inputs. The resulting DeepCGR achieves significant performance. DeepCGR can also predict expression levels of almost all genes by using only protein sequences without any information on the function or structure of the gene, and yet it shows good performance. DeepCGR can be an important computational platform that can be applied to various transcriptome data that measure the expression levels of genes in response to externally provided molecules. This study has been conducted with several people in the lab. In particular, I'd like to thank Professor Kim for his great help in the direction of the research. We are also very much grateful to the funding agency. Thank you very much for the kind attention.
Fantastic. Thank you for a great talk and video. So we have a couple of questions coming in through the chat. So first, in training the DeepCGR, did you consider other drug concentrations beyond the 10 micromolar?
Sorry.
Can you hear me?
Yeah. Sorry. Okay. I just noticed that what inference can you make to affinity potency of off-target effects of a compound?
Yeah. So I was reading through a different question first, which has been ticked off the list, which was, did you try and train the drug on train the model on different drug concentration?
Drug concentration. Okay, that's a great question. But when we dig into the L1000 database, there are, the more than 50% of the drug concentration, which I can deal with, was 10 micromole. So I didn't use any other things, but there are a lot of concentrations other than 10 micromole. So maybe we can use the other ones.
What's your related question? What's your thought on how concentration specific your model will be then in terms of the potency against the target and any off targets that you might be modeling?
So I cannot. Can you speak once again? Sorry.
Sure. If it's easier for you to read the questions, we can do it that way. But I was following on from the previous question to ask, how concentration specific, your model will be as a result of being trained just on that one concentration, and whether the potency of a compound and the specificity of a compound are obviously then going to affect the results.
Frankly speaking, I didn't thought about this kind of questions more, but I think that this concentration is not that specific and dependent on target expression level. Yeah.
Great. There's a question relating to the difference in predictions of the up and down regulated genes. So was there an imbalance in the original data set in terms of the numbers of genes that were up and down regulated by the drugs?
Yeah, that's a great question. So as far as I understand, the question is about how we deal with the imbalanced data set. So I think it is a kind of art to determine the percentage of upper and lower z-score as upregulation or downregulation. So there may not be a correct answer here, but as a result of referencing some previously published classification papers, so it was tentatively decided as 5%. So it's highly imbalanced to 5 to 95%. So we didn't do any other kind of oversampling or undersampling techniques. So the recall value is not that high as the other imbalanced data sets. So yeah, that's.
And a question around the poor performing compounds. So did you see any bias for certain chemical properties or mechanistic properties of the drugs for which the compounds for which the model performed poorly?
I'm really sorry. So can you speak once again? My audio is
It's
not
okay.
good. Yeah.
For the compounds for which the model performed poorly, were there any chemical properties or mechanistic properties biased towards those compounds?
So as if I understand well, so it is let's say so it's really it's highly depend on the kind of cell lines, but we distinguish that some set for each cell lines there are significant differences in the classification results, which upregulates or downregulates in terms of compound. So I cannot explain for just one or two cell lines, but yeah, I can say there are trends in result.
Great. Okay, thank you. There's several other questions in the Slido if you want to perhaps respond to them manually, but time now to move on to our next video. So thank you very much. So our next speaker is Dr. Edgar Kozlova, who's a postdoctoral researcher in the Department of Genetics and Genomics at the Icahn School of Medicine at Mount Sinai. He'll be presenting their work with single-cell sequencing of T cells to uncover signatures and regulators of immune cell tolerance to tumor vaccines. So over to Dr. Kozlova's video.
Greetings. My name is Edgar and I am a postdoc at LOSIX Lab. And I'm happy to present today for you the project titled Identifying Tumor Antigen-Specific CD4 T-cell Dysfunctional States by Single-Cell Transcriptomics in Immunotherapy-Treated Cancer Patients. But first, some disclosures. The immune system is composed by several populations of cells that switch between activation and suppression states, mostly to have a proper reaction towards harmful antigens and pathogens and being non-responsive towards the self, as shown on the left screen. On the right panel, we can see that the main effectors of this response are CD4 positive T cells that are going to be producing the signals to stimulate other immune cells like CD8 and myeloid cells, which makes them critical for the immune system. To study tolerization, a set of patients was subjected to several cycles of immunization. As seen above, each cycle contains multiple vaccinations. In blue, we show the humoral and cellular responses after receiving an initial vaccination without adjuvant. This produces a poor co-stimulatory environment, leading to a lack of antibody and cytokine production. Even after introducing adjuvant later, the responses are locked. The condition shown in green corresponds to a common immune response with proper antibody and cytokine levels. With this starting point, now we can focus on our objective, that is identifying the transcriptional program behind these tolerized antigen-specific CD4 T cells. And for this, we are going to be studying as a strategy the tumor antigen-specific T cells from before and after a vaccine immunotherapy under these two settings. The experimental design consists in raw blood of these patients before and after antigen vaccination. This figure illustrates over time in red the vaccinations and in black the sampling points. The color in each circle reflect which conditions were sampled. This next slide shows the sample processing, where we do initially a sorting to purify the T cells, then these are expanded, then they are stimulated with the antigen, then they are sorted again based on this stimulation, and finally extracted the RNA. After the sequencing, we perform a differential expression and linear modeling analysis to derive the immune signatures belonging to each class of CD4 T cells. The first signature I'm going to show you is the tolerized signature. This is the figure, the heat map for the respective genes that we included in this signature. And on the right side, we can see the PCA based on this immune signature. We can see that the groups are clearly separated except for that middle point called ECO1, which was a patient that had pre-existing immunity, and therefore it was behaving like one of the other conditions. In the next slide, we can see the same type of analysis, but this time we used known genes associated with CD4 cell exhaustion, and we can see that the profiles are not as clear as before in the heat map, and even more so when we see the PCA, we see that there is no distinction between the tolerized and the baseline group for these exhaustion-associated signatures, meaning that this is an entirely different process compared to exhaustion. And now we're looking at the gene scores represented as a median of the gene expression for the genes belonging to each set. For the tolerized score, we can see that the tolerized group had a much higher signature, reaching its maximum at 4 weeks, and for the immunized group, this signature was much lower. Both signatures were higher compared to baseline, and this is quite interesting because tolerized group is also showing a high variation between the patients for this signature. On the other hand, the T-cell exhaustion score on the right is showing that this is a continuous increase for the immunized group over time, where it seems to be also increasing over time for the tolerized group. It's not distinguishable from the baseline, which is shown in red. The shades represent the variation for the samples, and we can see the tolerized and immunized signatures for exhaustion are highly overlapping. Now we are looking at the Gini index representing how diverse these cell populations are. The figure on the left demonstrates that the immunized group suffers a loss in diversity, while the tolerized patients remain highly diverse, which is similar to baseline. However, the immunized group also shows a high variation for clonal diversity between patients, seen by the extend shadow in week 4. On the right side, we observe that antigen stimulation also remains low for the tolerized group, which also a very large variation between patients seen by the overlap of the shadows with the immunized group. Single-cell sequencing allows us to explore the expression profiles for each cell within each sample. The Disney plot allows us to get a sense of the expression profiles per cell. Here on the left side, representing each cell, and illustrates how different these three conditions are. Meanwhile, on the right side, we observe these cells colored by patient, illustrating how unique each sample behavior is at the expression level. These gene signatures identified here were similar to the bulk sequencing, but now we can draw carefully with much more detail of the specific gene expression profiles between cells within each patient sample. Because of the surprisingly high heterogeneity at the gene expression level, we decided to take advantage of an algorithm to quantify transcription factor activation status. Because these transcription factors are upstream of expression, we were expecting a much cleaner picture. For this, we calculated the profiles for each cell in the experiment and obtained a binary map of on and off for a set of 110 transcriptor factors. These profiles then revealed the different on and on switches for the tolerized and immunized cells, showed here in red and blue. Later, we performed a Disney analysis for the transcription factors, and we observed a very similar profile to the single cell map, but with much defined clusters. If we median the transcription profiles to obtain a score per condition, we can accurately identify which cells are tolerized, showed here in the top left blue, or immunized, shown on the top right red. Moreover, we can do a quick logistic regression between the TF profiles and obtain an accuracy of around A and an area under the curve, or AUC, also of 0.8, when classifying these cells. The bottom plots, left and right, show these profiles colored by combined scores and per patient respectively. Although we obtained a much cleaner picture upstream of expression, we were not satisfied, and we are currently observing the methylation profiles for these cells. Finally, I would like to thank Dr. Losik from the Department of Genetics and Genomics for his mentoring, Dr. Sasha Gnatic for this outstanding project, Shingo and Naoko for generating and producing results prior to my arrival to the team, and of course, this was the work of many amazing people that are not showed here. Thank you for listening, and I'm happy to answer some questions.
All right, great. We already have a few questions. The first question is, which genes or pathways are up or down regulated in the tolerized state?
Hi, thank you for the question. We have seen a set of 30 to 40 genes that are combined between down and up regulating for the tolerized state. However, we are still deciding on which pathways are the most relevant. And this is why we are combining single cell and bulk RNA-seq. And additionally, we are recently completing methylation analysis to verify some of these observations. Some of them are known markers, for example, LAG3, that is involved also in exhaustion, but there are some genes that are involved in Hitchcock proteins and others that are involved as well in metabolism, like GADPH.
Was there any difference in RNA-seq and single-cell RNA-seq results between the tolerized and exhaustive data sets?
Yes, we do have difference between the two analyses, but we also have some overlaps between them. Most of the differences are driven by the patient-specific variability.
And did you ever see whether tolerized gene sets could revert back to non-tolerized transcriptional states?
We haven't tried that experimentally yet, but we are planning for that soon.
Is the tolerized state unique to CD4 T cells, or do you see any indication that it would also apply to CD8 positive T cells?
So the reason for looking first at CD4 T cells is because they are going to be secreting the cytokines for inducing CD8 stimulation. We have not looked at CD8 T cells, but now we have reasons to believe that it may also be happening in CD8 cells.
How did you perform the transcription factor analysis in the transcriptomic data?
the TF factor analysis was only possible in the single cell data set, and we used a correlation network-based method called CINIC, which is publicly available.
And can you comment on how well the transcription factor profiles separated the individual patients?
Yes, we saw some. So this method allows us to quantify activation based on a much larger set of genes for each transcription factor. And we saw partial activation of the gene sets belonging to each transcriptor factor along the patients. So that means that we are still thinking how to interpret this, but some of these TF activation are partially activated in some patients, whereas fully activated in others. And that was the main difference between the patients.
Okay, great. And it appears that tolerization and immunization vary at the level of donor. Was there any heterogeneity of tolerized versus immunized within subject?
Yes, we saw we have some observations that there is some degree of heterogeneity. We haven't quantified that yet, though. But that's a great question, and that's something we can look for. Thank you.
Great. Thank you for a great talk. At this time, I will be introducing our next speaker, Dr. Tali Mazer, who is a computational biology scientist in the Knowledge Systems Group at Dana-Farber Cancer Institute. She will be presenting MatchMiner, an open source platform to match patients to clinical trials by clinical and genome eligibility criteria. So let's begin the presentation.
I'd like to begin by thanking the organizers for the opportunity to speak with you today. My name is Tali Mazer, and I'm a scientist in the Knowledge Systems Group at Dana-Farber. We're a diverse group dedicated to building software to support cancer care and cancer research. Today, I'd like to tell you about one of these clinically focused projects, a software platform called MatchMiner. I have no disclosures. The idea behind MatchMiner is fairly simple. Genomic sequencing of patient tumors is increasingly common. There are also an ever-growing number of clinical trials looking for patients with specific genomic features. With all this genomic data and all these trials, it can be hard to find the right patient for the right trial at the right time. MatchMiner sets out to bring these two pieces together. How can we connect patients and trials? We built MatchMiner with two main goals in mind: accelerating trial enrollments to cancer precision medicine trials, and maximizing trial options for every patient. MatchMiner connects patients to trials in two ways. In patient-centric mode, a provider can look up a specific patient and view genomically driven trial options for that patient. In trial-centric mode, anyone involved in running a trial can identify patients with specific clinical and genomic features who may be eligible for that trial. Let me show you what that looks like in MatchMiner. Here's an example of what you see when looking up a specific patient. You can see all the trial matches listed out, including the reason for the match. So here we're looking at a tumor sample with an IDH1 mutation, and that mutation is leading to these two trial matches. We can see information about the trial, and over on the right, you can click the envelope icon to directly email the study team. You can also click on the trial to see more information about it, and I'll show you that in a moment. First though, let's take a look at what trial-centric mode looks like. After you've created what we call a filter, which defines the clinical and genomic criteria of interest, you'll see a page like this, which lists out all the patients that currently match to your filter. You can click on a patient to see more information, and over on the right is a link to directly email the patient's provider if you think they're a promising candidate. Up on the top, we provide some bins to help you organize patients based on their eligibility. Finally, this is what you'll see when you look at a trial. You can access this page, as we saw, from a patient page, or MatchMiner has a standalone search feature, so you can always search for and filter trials based on a variety of genomic and clinical features. Here, we see just the top of the trials page. There's a lot more information available as you scroll down, but this is going to give you a sense for what's there. I'd like to take a step back now and talk about how we find these matches. In order to identify a patient trial match, there are certain types of data that we need. And because we're doing computational matching, we need that data to be in a structured format. Generally speaking, we can think about this as patient data and trial data. Patient data includes genomic data, and for us, that comes from two different targeted sequencing panels being run at Dana-Farber. We have OncoPanel, generally used for solid tumors, and the Rapid Heme panel for heme malignancies. Patient data also includes clinical and demographic data, things like gender or cancer type. In terms of trial data, we connect to our clinical trial management system for real-time information about which trials are open or closed. This enables us to only show matches to trials that are currently enrolling. However, there's one key piece of information that we haven't discussed yet, and that's trial eligibility. What are the specific inclusion and exclusion criteria for each trial? We obviously need this to successfully match patients to trials. This information is generally found as free text somewhere in a trial's protocol. So how can we transform that into a structured format amenable to computational matching? To address this, we developed Clinical Trial Markup Language, or CTML. This is a structured format to encode detailed information about a trial. It's also a flexible format that can encode a wide range of trial-related information. But most relevant for trial matching, CTML utilizes Boolean logic to encode inclusion and exclusion criteria. Importantly, the Boolean logic allows for the encoding of simple or highly complex criteria. And CTML allows curations at the arm, step, or dose level to accommodate any trial design. Currently, CTML supports a variety of genomic, clinical, and demographic data types, as listed here. But as I mentioned, CTML is a flexible format and can easily be extended to support any relevant data type of interest. Here's a simplified example of CTML. At the top, we can see trial information, the title, NCT number, whether the trial as a whole is open or closed. Below that is the treatment list, which is a tree-like structure of all arm, steps, and doses of the trial. Each level has its own status, since even an open trial may have some arms or doses that are closed. At any level, we can also add a match clause, where eligibility is encoded via Boolean logic. I'll show you an example of that on the next slide, but I first want to point out that by having status and curation at any level of a trial, we can provide precise trial matches based on the true state and requirements of a given trial. So what does that Boolean logic look like? Let's take a simple example. Here's an arm of a trial looking for KRAS mutation or KRAS amplification in any solid cancer type except breast. Here's what that looks like, and let's go through this step by step. We see here KRAS mutation, KRAS amplification listed out and connected by an OR to say that the trial requires either of these. Below that, we see the cancer type information, solid, not breast, and these are connected by an AND to say that both are required. The cancer type must be a solid tumor type, and it must not be a breast cancer. Finally, the genomic and clinical clauses are linked by an AND to say that in order to match, a patient must have either KRAS mutation or amplification and a solid tumor which is not breast. So now we can put that all together and see how that looks within the full CTML. Now earlier, I showed you part of the trial's details page. As you scroll down on that page, you're actually able to see this curation. And so here's what this same curation looks like in MatchMiner. This, of course, is a simple example. We can also encode much more complex criteria. For example, here we've curated for any mutation, but we could instead curate for a specific mutation, like only KRAS G12D mutations. Here we see an example of a clinical exclusion, but we could also exclude genomic features. At this point, you can hopefully appreciate that this simple Boolean logic can be used to encode any level of complexity in a trial. I'd like now to discuss a bit about the technical details of MatchMiner. MatchMiner is an open source project. All the code is available in our GitHub repo. MatchMiner is a two-tier web application with a Python-based REST API and an Angular front end. It uses SAML-based authentication, and when hosted behind an institutional firewall, is fully HIPAA compliant. Importantly, MatchMiner can connect to existing clinical systems. For example, to read real-time trial status from a clinical trial management system, or to an electronic health record for ease of access within a clinical workflow. In the schematic on the right, we can see all the different data inputs that are joined together by the match engine. This is one of the key features of MatchMiner. It's the core matching algorithm which takes in all the patient and trial data and identifies matches between the two. We recently refactored this core matching functionality to improve it in various ways. It provides highly detailed reporting of the reason for each match. It can be run in multiple different ways. You can run all patients against all trials, or only living patients against open trials, or individual patients or trials, depending on your needs. Finally, we've made the engine easily extensible for new data types. Rather than needing to edit the core code, the majority of data type-related customizations are available as configurations. Now that I've introduced you to the platform, let me tell you a bit about how we've used MatchMiner at Dana-Farber. We launched a preliminary version in 2016 and have been fully operational since 2017. We curate any trial that opens at Dana-Farber with genomic eligibility, and since launching MatchMiner, we've curated more than 300 trials. We also have genomic sequencing data for more than 37,000 patients. What does that mean for an individual patient? Well, more than 80% of patients have at least one trial match, and on average, patients have 6 trial matches. And to date, we're aware of more than 115 patients who've enrolled onto a clinical trial as a result of MatchMiner. You can see a graph of that plotted on the right. As I mentioned, we've been fully operational at Dana-Farber since 2017, and MatchMiner is regularly used by clinicians across all disease centers. In order to continually improve MatchMiner, to identify opportunities to further develop the system, we have multiple collaborations with clinical groups at Dana-Farber, including supporting trial investigators in finding patients, supporting molecular tumor boards, and identifying standard of care precision medicine options. It's feedback from these collaborations and from users in general that drives the development of MatchMiner. In addition, this is an open source project, and there is a growing community outside of Dana-Farber. We're aware of at least 5 clinical centers who are implementing MatchMiner locally. We're committed to continuing to develop MatchMiner based on feedback from all of these sources. Finally, MatchMiner is a team effort, and I would like to end by acknowledging and thanking all the current and former members of the MatchMiner team, as well as our funding sources. If you're interested in learning more, please visit our GitHub repo, read our documentation, or go ahead and email us. We'd love to hear from you. Thank you for your attention.
Great, thank you for an excellent talk and an excellent video. So there's a few questions coming in through the chat for Dr. Meza. So we'll start with within the CTML language and the matching. How do you deal with the hierarchical nature of some of these terms? For example, avelumab or durvalumab sitting underneath PD-L1 inhibitor, sitting underneath immunotherapy, etc.
Yeah, that's a great question. So within a specific trial, we'll kind of have the information, what drug is the trial actually using. We don't have that sort of hierarchy built into the system at this point. So when searching for a trial, you can search by drug, and you would need to kind of search by each individual immunotherapy. It is something we've thought a lot about and would like to include at some point in the future, would be to be able to, instead of searching for the specific immunotherapy drug, search for any trial treating with an immunotherapy drug, but we don't have that yet.
Great, thank you. And is the trial or the patient data curation done manually, or can that be automated in some way, using something like natural language processing?
So right now we're doing it manually. It certainly could be automated, but what we're doing right now is we get essentially a scaffold of a trial from our clinical trial management system. So a lot of the information is there, and then we just fill in the actual eligibility, and we are doing that manually. It is certainly a time-consuming process, but at this point we find that it kind of gives us the best, most accurate representation of what the true eligibility is.
Great, and then a couple of questions coming in around instances where patients match to multiple trials. So is there a prioritization process or decision support to help identify which trial would be the optimal?
Absolutely, that's an important question and often a hard thing to figure out. So automated within the system, there is a prioritization, and it's based on a couple different things. It's based on, in part, some of the data from our sequencing assays that we're using. So when those reports are signed out, mutations are given a tier or a pathogenicity. And so we incorporate that information. If a variant was signed out as pathogenic or as kind of a high tier likely to be relevant, those trial matches based on those variants will be ranked higher than those based on what are essentially variants of unknown significance. And in addition, we'll take into account how specific the trial was in their criteria. So a trial that's looking for a very specific mutation, if a patient has that same mutation, then that is a higher ranked match than a trial that's looking for any mutation in the same gene, because the trial kind of included that specificity. So we do it in that way. And we also have some collaborations with clinical groups where we try to add in kind of a level of human interpretation on top of that as well, and have essentially a molecular tumor board report for some patients that, given the conversation among various experts, how do we rank the trial matches? And so we're doing that. That obviously is much more time intensive, and so done on a much smaller scale, but we're certainly thinking about ways we can integrate that more broadly.
Great. A couple of questions around, ease of access to this from people outside of the system. So, you mentioned it was open source, but thinking about kind of how easy it is for a patient or an investigator to push data onto the CTML format and really kind of take advantage of it.
Yeah, great question. So right now, the way that the system is set up, it is kind of internal to an institution. So it's hosted behind our firewall at Dana-Farber. That's part of how we keep it HIPAA compliant. And so at this point, our instance at Dana-Farber is really only available to Dana-Farber people. Instances at other institutions would similarly be only available kind of to people at that institution. For anyone at our institution, we can work with them in terms of getting their data into the system as needed. We are, though, also starting to think about and have conversations about how can we make this available a bit more broadly, because it is a fair amount of work to get it set up at an institution. And so we're starting to think about how we could potentially work with other hospitals in our community who might have patients that could easily come to Dana-Farber for the trial. How can we kind of integrate some information, and how can we better support the other hospitals in our area? So we're having a lot of conversations about that, but nothing kind of set there yet.
Super. Thank you very much. So there's some really great questions still remaining that unfortunately we don't have time to. So maybe you can go and have a look at those in the Q&A system. But thank you for a fantastic talk and the answers to those questions. And it's now time to move on to our next video from Dr. Jun Lee, who's an assistant professor in the Department of Bioinformatics and Computational Biology at the University of Texas MD Anderson Cancer Center. He'll be presenting Dr. OncoWrite, which is a chatbot that promises to democratize cancer omics, enabling use of natural language to explore and analyze cancer genomic data sets. So over to the video.
Hello everyone, my name is Jun Lee from the Department of Bioinformatics and Computational Biology at MD Anderson Cancer Center. Today, my talk is about a new bioinformatics platform we have developed, which is a natural language-oriented analytics platform for cancer omics data. I have no financial relationships to disclose. As you know, over the past decade, high-throughput molecular profiling technologies have revolutionized cancer research. Petabytes of omics data have been generated from thousands of patients and cell line samples, especially through some large consortium projects such as TCGA and ICGC projects. These high-throughput cancer omics data have provided a great opportunity to characterize cancer-related molecular mechanisms and identify biomarkers and therapeutic targets systematically. But this high-dimensional molecular data also presents a major challenge for cancer researchers in analyzing the data and obtaining meaningful biological and clinical insights effectively. This is particularly true for cancer researchers who have no or limited bioinformatics and statistical expertise. As a result, there is still a substantial barrier that prevents a large body of cancer researchers from performing cancer omics data analysis in an intuitive, efficient, and reproducible way. To address this challenge, our team has developed Dr. OncoWrite, an open-access, natural language-oriented, artificial intelligence-driven analytic platform. As you can see from this snapshot, a major feature of this platform is that it allows users to ask biological questions through natural language directly. It then automatically translates users' intentions, identifies the related cancer genomic datasets, performs diverse bioinformatics analysis, and returns the results in a timely, visually attractive way. As shown in this workflow, through an online chat interface, users can simply type an omics data analysis question in the input area. After receiving the input text, the program will run its NLP-based module to tag the recognized entities based on the identified features. The backend AI module will calculate scores to predict the best matched analytic task. The program will then call the analytic module, identify the related dataset, and check if all required parameters are good. Before submitting the task, Dr. Uncle Rat will send a message to the user to confirm if the detected task is the user's intended analysis. If confirmed, a job scheduler will submit it to a job queue and use cloud-based compute nodes to process the job. Once the job is finished, Dr. Uncle Rat will call the corresponding visualization module. Finally, the results will be sent back to the user through the dialog area. Importantly, Dr. Uncle Rat provides a rating feature for each successfully executed job. The feedback received from each task will then be used as the training data to improve the performance of the NLP and AI models. Now let's look at a few examples. This is the user interface of Dr. Uncle Rat. You can ask questions at the bottom input area. For example, how many cancer types do you have in TCGA? Dr. Uncle Rat will confirm with you about what you would like to do. Then you need to input yes to start this task. Now you can see bar plots to display all the cancer types with the distribution of number of samples. So how about the correlation analysis between two genes in TCGA breast cancer? And it asks you about what type are you looking for, and we input gene expression and confirm, and you can get a scatter plot with p-value and correlation coefficients, and you can also download the plot and rate for this analysis. So how about differential analysis on a gene expression and gender in TCGA kidney cancer? You confirm and you get the box plots with the p-value, and you could download the plots and rate the analysis. So let's look at another example. Please do an overall survival analysis on TP53 gene expression in breast cancer. So it asks you about the data source you're looking for, and we input DCGA. Here you will get a survival plot with the log-rank test p-value and Cox p-value. Another example is to show the lollipop plot of TP53 mutation in TCGA kidney cancer. And this is a lollipop plot for TP53. You can see the distribution of mutations in different domains. Let's rate. And say thank you to Dr. Rat. Let's look at what kind of analytic modules that Dr. Uncle Rat has. From this figure, you can see these modules cover all major directions in genomic analysis, including analytic tools, pipelines based on different molecular data types, complex heat maps, network pathway analysis, single-cell analysis, pan-cancer analysis, and commonly used statistical analysis. As you know, reproducibility is a major concern in biomedical research. Dr. UncleRed provides an extremely convenient way to check the reproducibility of some published genomic analysis. To demonstrate this point, we focused on a classic cancer genomics paper published in Nature, which has been cited by 1,000 times. This study includes some common cancer mutation analysis. After loading the published dataset from the paper, the key results in the main figures can be easily reproduced through quick dialogue-based queries. In addition, we keep adding new modules. As shown in this figure, we have already developed messaging analysis, user rating, speech-to-text, and authentication modules. Currently, we are working on other new features such as room chat to enable collaborations and group signs, smart demo to automatically guide users on learning our system, and the upload feature to allow users to analyze their own data. To maximize the utility of Dr. UncleRed, we are also developing mobile apps to enable users to use our Dr. UncleRed anywhere and anytime through the mobile phones. This is definitely a team science. Finally, I'd like to thank all the team members. To give my special thanks to Dr. Han Liang, who leads this project, also to our talented core members, Hu Chen, Yumeng Wang, and Meiru Chen for their great efforts, creative ideas, and enthusiasm in developing Dr. UncleRed. In addition, I would like to thank our department and IT groups for their great support on this project, especially our chair, Dr. John Weinstein. Thank you.
Another great talk. Thank you, Dr. Lee. We now have some time for questions. The first question is, which databases does Dr. UncleRed have access to?
So currently we have TCGA, ICGC, and GTEx and CCLE data created in our database, but we plan to add more during the development.
Great. Is there any data cleaning or harmonization performed for the aggregated data accessed by Dr. UncleRed?
Yes, we manually doing that for different data sources. We actually use our expertise for our pan-cancer analysis in TCGA and ICGC, and to standardize the data based on the standard procedure used in the public cohort data. Yeah.
And a related question, does Dr. UncleRed detect batch effects?
Currently, we do not have that, but we can do that in the future development, definitely, because many users, they would like to do the analysis across different data sets, and batch effects definitely a critical issue when you doing such analysis.
For overall survival, what is the default criteria for high versus low expression groups? And can we ask the AI to customize it?
Currently, we use the median value to characterize the samples into high group and low group, but we are trying to make it more customized version that the user could, for example, do the subtype analysis or define their own subgroups to do the survival analysis. Yeah.
Can it be plugged into any database with necessary modifications? And can you implement the workflow as a batch file?
Yes. So we currently use the standard way to process the data, but in the future we will use the workflow language to standardize the whole process and make it reproducible and open to the user, that they could use such open workflow language to reproduce everything we have done using Dr. UncleRed.
Can the code used to make the plots be accessed? Is it done with Python or R?
Again, it's similar to the last question. So
basically we currently generate a report for the sequencing data, and later we will use the report to include both the code and the results we have underlying the interface, and then the user could see the results as well as the scripts, the R scripts or Python scripts we have used. Then they could just apply the scripts in their customized data or their own analysis.
We have a couple of questions related to accessing the tool. Is there a particular browser requirement? Is it available now?
Yes, it's available now. And if you want to use all the features, the best one is the Chrome, but we are trying to make it comparable using the Safari. And so, yeah, that's the current stage.
Are you aware of any other tools like this?
No, I don't think so. I think the Dr. Ankurat is the only one using a dialogue NLP-based query form to do the genomic analysis.
And finally, can you comment on the strategy for query expansion?
Query expansion, let me see. So you mean the to improve the query, or?
The question doesn't provide any additional details. So
okay. So basically, we currently have the query in the interface, and then we process the user text. And then because as we accumulate more user query texts and the feedback from the user, then we could train the data based on the user query text and their ratings, as well as the functionalities we matched using the query text. Then we can keep improving the whole process. And then I think we can do better and better based on more user query text pool we accumulated from the backend database. Yeah.
Thank you so much. There's a few additional questions that we'll post to the chat. And at this time, I'll go ahead and switch to the concluding statements.
Okay, thank you.
So technological advances in generation of biological measurements and advances in bioinformatics and AI methods capable of inference from high-dimensional noisy data have set the stage for new discoveries and innovations to benefit cancer patients. Today, we heard about 7 really exciting examples in this space. Ms. Shi and colleagues combined single-cell sequencing and bioinformatic analysis to uncover differences in the airway field of NSCLC patients with adenocarcinoma versus squamous cell carcinoma. Dr. Deganaziri described a new algorithm called Sicilian to profile gene fusions from single-cell RNA sequencing data. Dr. Adesmaki described a computational repurposing of public cancer databases, including the TCGA and the DepMap, to streamline intelligent drug target discovery. Mr. John told us about the application of deep learning to predict gene expression responses to small molecule perturbations in cancer cell lines. Dr. Kozlova and colleagues combined single-cell sequencing with bioinformatic analysis to uncover a new signature of T-cell tolerization, a new form of T-cell dysfunction in CD4 T-cells. Dr. Mazur described MatchMiner, a tool to intelligently automate the challenging task of matching cancer patients effectively to clinical trials. And Dr. Lee just told us about Dr. Onkerite, an AI platform that makes bioinformatic analysis of existing cancer data sets more widely available to cancer researchers. These are all very exciting developments. In particular, the talks by Dr. Adesmaki and Dr. Lee talked about applications that use or take advantage of existing publicly available cancer data sets. The opportunity to ingest increasingly large data sets to power biomedical discovery is incredibly exciting. Notably, data generation and sharing remain at the heart of our ability to perform bioinformatics and AI for applications in cancer research. To To realize the full promise of this growing area, it's important as a community that we must promote fair data sharing principles, namely making data findable, accessible, interoperable, and reusable, so that individuals performing bioinformatic and machine learning can most effectively take advantage of these data. Another important consideration is the bias implicit in available data sets, as the tools we train can only learn from the patterns in the data that they are presented with. Employing strategies to detect and remove bias from machine learning should become standard practice and will be particularly important for tools used in clinical decision-making. Finally, I'd like to thank our speakers once again, as well as everyone who submitted abstracts to the session and everyone who is here participating with us today to make this such a fantastic session. And thanks once again to the organizers for making everything possible. This now ends our mini symposium on bioinformatics and artificial intelligence for cancer research and development. Please stay well and enjoy the rest of the meeting.